Welcome to the deep dive. Today we're jumping into some really fascinating developments in prostate cancer research. We're looking at stuff that's hitting the clinic now, but also some, well, pretty groundbreaking molecular work happening behind the scenes. It feels like a really pivotal moment for the field.
It absolutely is, yeah. Our goal here really is to connect the dots between these recent studies. We want to show you how researchers are kind of fighting on two fronts. First, improving the treatments we already have, making them work better and for more people today. And second, developing these completely new strategies designed to outsmart resistance before it even starts.
Okay, so we've broken this down for you. First up, we'll tackle the clinical side stuff impacting patients right now. Then we'll shift gears to the really innovative preclinical science, the things that could totally change the game down the road. Let's kick off with something impacting a lot of patients, that ARASENS subgroup analysis.
Right, ARASENS. So this looked at that triplet therapy, darolutamide plus ADT, androgen deprivation therapy, and docetaxel chemo. This is for men with metastatic hormone-sensitive prostate cancer, MHSPC. The main trial showed it worked, but the big question, the one doctors always grapple with, was, does it work as well in older guys? 75 plus.
Yeah, that's always the worry, isn't it? More side effects?
Yeah.
Maybe they can't tolerate such an intensive combo.
Exactly. There's often hesitation. So this analysis split the patients under 75 and 75 or older. And what they found was, frankly, quite striking. The survival benefit held up. Actually, it looked even a bit better in the older group.
Okay, let's put some numbers on that. Under 75.
About a 30% reduction in the risk of death with the triplet.
And for the 75 and older group.
Get this, a 39% reduction, almost 40%.
Wow. That's huge. That really pushes back against the idea that older patients can't benefit from intensive therapy.
It absolutely does. It's a practice-changing finding. And it wasn't just survival. Progression was delayed massively, too. The median time doubled, basically. Went from like 19 months without darolutamide to over 40 months with it. And importantly, the safety profile was manageable, consistent across both age groups. It really cements this triplet as a standard of care, regardless of age.
Okay, so better survival is key, but treatment itself can be tough. Let's talk about that side effect everyone dreads with ADT, fatigue. It's not just being tired. It really hits quality of life, makes it hard to stick with treatment sometimes, a big unmet need.
Oh, absolutely. It's pervasive. So this next piece looks at a different angle. A phase 2 trial out of the University of Arkansas is testing low-dose naltrexone, LDN, just 3 milligrams taken nightly.
LDN, naltrexone. Isn't that usually for opioid issues? How does that connect to ADT fatigue?
Yeah, it seems counterintuitive at first, right? But the thinking has shifted. They're targeting men on ADT, either alone or with the newer drugs like apalutamide and xelutamide, those types. And the rationale is that ADT seems to cause this low-grade chronic inflammation. You see levels of things like IL-6, interleukin-6 go up.
So it's not just physical exhaustion, it's potentially a immune or inflammatory response to the hormone therapy.
Exactly. That pro-inflammatory state seems tightly linked to how bad the fatigue gets. And LDN at these low doses seems to have immunomodulatory effects. It can potentially dampen down that inflammation, reduce those cytokines.
Okay, so if it's inflammation, they need to measure more than just how tired patients say they feel, right?
For sure. The main outcome is patient-reported fatigue using the ECA-CF scale. That's key. But crucially, they're also measuring biomarkers, looking at mitochondrial function, inflammation markers, oxidative stress. They want to see if the drug is actually hitting that biological pathway and making people feel better. It connects the mechanism to the symptom.
All right. Fascinating approach to a common problem. Now let's pivot. We've covered refining current care. Let's look ahead. Let's move into these newer platforms emerging from the lab, starting with something called GenSci-143. It's a dual-targeting ADC, antibody drug conjugate.
Right. We're stepping into the realm of molecular precision strikes now. ADCs are like guided missiles. An antibody guides a chemo payload right to the cancer cell. Very powerful. But a common problem, especially in metastatic castration-resistant prostate cancer, mCRPC, is tumor heterogeneity.
Meaning the cancer cells aren't all the same. Some have the target, some don't.
Exactly that. If your ADC only targets one protein, say PSMA, but some cells don't express much PSMA, those cells survive, grow, and your treatment stops working. That's antigen escape.
So GenSci-143 tries to get around that by targeting two things at once, like a backup plan.
That's the idea. It targets both B7H3, that's CD276, and PSMA. By hitting two different markers, often found on prostate cancer cells, you significantly increase the odds that the ADC will bind and deliver its payload, even if expression levels vary.
And this one seems to be moving fast. IND accepted by the FDA, trial approval in China back in October 2025. It's heading straight into phase 1 trials.
Yeah, the preclinical data must have been pretty compelling. In lab models, these cell-derived xenografts, it reportedly outperformed similar single-target ADCs like DAS 7300 or ARX 517. So the dual targeting strategy looks promising, at least preclinically, for dealing with that heterogeneity issue.
Okay, sticking with advanced engineering, KRRT cells. Huge success in blood cancers, but solid tumors like prostate cancer have been, well, a tougher nut to crack. What's AB 3028 doing differently?
Right. CRRT for solid tumors faces several big hurdles. One is that same heterogeneity we just discussed, finding a target on all the cancer cells. Two is off-tumor toxicity, accidentally hitting healthy cells that might share one of the targets. And three, maybe the biggest one, is the tumor microenvironment, the TME. It's incredibly suppressive. It actively shuts down T cells. TGF-beta is a major player there.
So AB 3028 is designed to tackle all of that. How? You
Yeah.
mentioned sequential AND logic.
Yeah, this is where it gets really clever. It's engineered with what they call an AND logic gate. Think of it like needing two keys to unlock something. The CRT cell has to recognize two different antigens on the cancer cell surface before it gets fully activated to kill.
Okay, like two-factor authentication for cell killing.
Hmm.
It sees antigen A but waits, needs to see antigen B as well before unleashing the attack.
That's a perfect analogy. It dramatically increases specificity. If a healthy cell only has antigen A, the CRT might bind weakly but won't launch the full attack. It reserves maximum power for cells showing both signals the tumor signature. This should really reduce off-tumor toxicity.
And what about fighting back against that suppressive TME, the TGF-beta problem?
It has another built-in trick, an shRNA mirror module, basically molecular machinery inside the CRT cell. This module acts like noise-canceling headphones against the TGF-beta signal. It actively knocks down the receptors that TGF-beta uses to tell the T cell to shut down specifically FAS and TGF-beta R2. So the CRT cell becomes resistant, it can ignore those stop signals and keep working even inside that hostile TME.
Wow, so precision targeting plus built-in resistance to suppression, that sounds like a serious step up. And the early data backs up the ANI-gate concept.
Yeah, preclinical tests showed it worked as designed, strong killing of cells with both antigens, minimal effect on cells with only one. It's the kind of smart engineering we need for solid tumors.
Okay, one last major finding, and this one feels really fundamental, going right to the core of hormone resistance. A study in Nature identified something they called a new weak spot, a protein called PTGES3, or P23.
Right, so we know the androgen receptor, AR, is the main driver of prostate cancer growth. Most therapies try to block it or reduce androgens, but cancer finds ways around that. PTGES3 seems to be a crucial helper protein, like a chaperone or scaffold. It helps keep the AR protein stable and active, even when hormone therapies are trying to shut it down. It allows the cancer cell to kind of maintain that AR signal.
So we're trying to block the main switch, AR, but the cancer uses this PTGES3 helper to keep the switch working anyway.
That appears to be the mechanism, yes. They used CRISPR gene editing to screen for genes essential for AF function, and PTGES3 stood out. When they switched off PTGES3 in lab dishes, the AR-driven cancer cells stopped growing, and many actually died.
Oh, that's in a dish. Did it hold up in animal models?
It did. In mouse models, reducing PTGES3 slowed tumor growth and, interestingly, also lowered the amount of the AR protein itself within the tumors. This suggests it's truly functional, not just correlated. It's needed for that resistance phenotype.
And the clinical link, does this PTGES3 level matter in actual patients?
Yes, and this is critical. They found that higher levels of PTGES3 in tumor samples from patients correlated strongly with worse outcomes, faster progression, poorer survival, specifically for patients on standard hormone therapies. It looks like a key resistance factor we weren't even measuring before.
So the implications are testing for PTGES3 levels to predict resistance, and maybe developing drugs that target PTGES3 itself.
Exactly, both. First, it could become a biomarker to identify patients likely to fail hormone therapy early. Second, and maybe more excitingly, it offers a completely new non-hormonal target. You could potentially develop drugs against PTGES3 to use alongside existing hormone therapies, maybe resensitizing resistant tumors.
Okay, let's try and pull this all together. We started with confirming a powerful triplet therapy works well even for older patients, which is immediately impactful. Then look at tackling a really common side effect, ADT fatigue, through an anti-inflammatory approach with LDN.
hmm And then we shifted to the future. These next-gen platforms like dual-targeting ADCs, the GenC 143 trying to beat heterogeneity, then the really sophisticated CAR T cells, AB 3028 with that AND gate logic and TME resistance built in, and finally, digging down to a fundamental resistance mechanism by identifying PTGS3 as a key AR helper protein, a totally new therapeutic angle.
It really paints a picture of a field advancing on multiple fronts simultaneously, doesn't it? Improving today's care while engineering tomorrow's precision cures. We're moving beyond just broad hormone blockade to these really specific multi-pronged attacks designed to anticipate and overcome resistance.
That's the core message. It's about molecular precision, understanding the escape routes, and building therapies to block them. The synergy between optimizing current treatments and developing these highly targeted next-gen approaches is what's going to reshape outcomes.
It's an incredibly exciting time. Lots to watch in the coming years.
Stay strong and fight on.